Hello students, welcome to HSC Chemistry and the acidic environment. This particular video is going to focus on buffers. So firstly, we need to understand what the concept of a buffer solution is. And the important thing about a buffer solution is that a buffer solution is something which can control changes in pH. So it's designed, it's an equilibrium system designed to resist changes in pH, but only over small quantities of acids and bases. Now, something like small obviously has some associated scale with it, but it's important that you remember that as far as buffer solutions are concerned, if you pour a large amount of concentrated acid into them, then obviously they won't function properly anymore. But as long as the fluctuations are reasonably small, then the buffer is very good at resisting changes in its pH, so it can create a stable pH. The buffer itself, the components of the buffer are going to depend on what type of pH we're trying to maintain, so we can have acid-based buffers, neutral buffers, or slightly basic buffer solutions. But usually a buffer solution is a mixture of an acid, which is a weak acid, and its conjugate base. Okay? Each of these two in roughly equal concentrations. So if we represent our acids the way we have in the past, HA, and this is in equilibrium, and you have H plus ions and A minus ions, then what we want is the concentration. If the concentration of this is X, then we pretty much want the concentration of this to be X as well. Now, obviously this opens up a few interesting little possibilities, given that if this is a weak acid, then it's not going to readily produce its ions. So therefore buffer solutions have to be combinations of both the weak acid and also a salt which produces the anions. And we'll have a look at some examples of this as we go along. So what we want to do then is we want to look at what happens to a buffer solution when we make some change, either by adding an acid or alkaline. So let's look at one particular type of system, which may be the acetate or ethanoate, COOH, which is in equilibrium with H plus ions and CH3 COO minus ions. Now, if this is a buffer solution, that doesn't look really good there. If this is a buffer solution, then we need to have roughly equal concentrations of our acid. This is our acid, and this is our conjugate base over here. Okay, so obviously we can add a source of acetic acid, but here we would probably add something like sodium acetate, because the sodium ions are going to remain in the solution and not affect the pH at all. So if an acid is added to our buffer solution, then our acid is going to increase the concentration of the H plus ions. So in order to remove these extra ions, the Le Chatelier principle tells us that the equilibrium is going to shift to the left, and it means that the excess acetate ions are going to combine with the excess hydrogen ions, and we're going to reform more of our molecule, our acid molecule, acetic acid over here. If a base is added to the buffer, then effectively what is going to happen, if I just quickly draw this equilibrium again, same thing. And if we assume that what we're doing is we're adding a source of hydroxide ions, what hydroxide ions are going to do is combine with the H plus ions to form water. So the H plus ions are going to remove the hydroxide ions, H plus, and that's going to mean that the concentration of hydrogen ions is going to decrease. Now that's the change. That's not the Le Chatelier shift, that's the change. The shift comes in trying to reverse the change. So in this case, if the concentration of the H plus ions has gone down, then the equilibrium is going to shift to the right in order to replace them. So we'll have a drop in our concentration of acetic acid molecules and an increase in the concentration of the acetate ions. The net effect of either of these changes is that pH of the solution contained in the buffer remains reasonably constant, okay, and hopefully should fluctuate within a small range. As I say, if it's a very strong acid solution that's being added, then it's a very large number of H+ ions, and that will make it very difficult for the buffer to resist those changes. But as long as the changes are small, the buffering system works very well. Buffers exist in a number of different situations in living organisms and also within the environment. One particular buffer system that exists in living cells is this one, this conjugate acid-base pair. So if we were to write this equation in, I guess, simple terms, then what you've got is a dihydrogen phosphate ion, which is in equilibrium with an H+ and the HPO4 2-. So this is acting as an acid and this is acting as a conjugate base. So how is this system capable of resisting changes that occur in the pH of living cells? Well, the easiest way to do that is to have a look at what happens in each situation when we are looking at the addition of, say, OH- ions here. If we add OH- ions, then this equilibrium is going to shift in such a way as to produce water molecules. Okay, so the OH is going to combine effectively with the H+, and we're going to increase the concentration of the HPO4 2- ions. Okay, so as we saw before, this is the species that's going to be attacked when we add base. If we add an acid, it's going to be the opposite one. Okay, HPO4 2- will combine with a source of hydrogen ions to likewise give us water in liquid form and the H2PO4- ion. So what we've shown is that in each case, one part of this buffer system up here is going to resist the addition of a base, and one part is going to resist the addition of an acid, and on each side of these equations you can see both species that are part of our buffer system. Now, buffers also occur in the blood, same sort of reasons, and I won't go through the equilibrium in quite the same way, but in the human blood we have a bicarbonate ion, and this one will combine with H+ or H3O+ in an equilibrium to form carbonic acid. Now this is a little bit more complex, mainly because we also have another equilibrium that exists in the blood. This equilibrium incidentally also exists in seawater. It's one of the ways that seawater is buffered against major changes in pH. But it's also useful in human blood. The problem with human blood is we also have some other things going on, such as carbon dioxide and water, and they too are going to change this particular equilibrium. So there are a couple of things that are happening. But again, if you look at these two species, then we get a bit of a sense of what happens as we add OH- ions. And we're going to have the acid. This one here is the base. This is the acid. In this case, the conjugate. And then obviously the base is going to take up any excess hydrogen ions to form the acid. So this one is likewise acting as a buffer solution because again we have two species in equal concentrations or roughly equal concentrations and an equilibrium existing between them that allows any changes that are made in terms of acidity to be counteracted as the equilibrium shifts. So here's a question from the 2012 HSC paper, question 8. It's, as you can see, a multiple choice question. Which acid-base pair could act as a buffer? So our answer to this question means we need to have an acid and its conjugate base. Okay, so we need to make sure that we have acid base. Do we have that for each one here? That's an acid base, that's an acid base, that's an acid base, that's an acid base. Okay, so all of them actually fit the category. We want a weak acid, so one of these already stands out as being not a weak acid. All right, nitric acid is a strong acid, and because it's a strong acid, it's no good. And these other two are an equilibrium involving water, and we know that water is particularly weak and not a very good choice as a and because water itself is weak, its ions are strong, so higher concentrations that we'll have in neutral solutions. We have equal concentrations of these, and this is not going to give us our standard definition of a buffer. So the only one that actually qualifies is the system that we looked at for living cells, which is the hydrogen phosphate equilibrium. So D is the correct answer. Thanks for watching.